In this video, we will discuss about sialic acid in pancreatic cancer. Cells can derive the tumor-associated macrophages differentiation with the help of this Siglec receptors, that is Siglec 7 and Siglec 9. So the first figure of this paper focus about the sialic acid on the pancreatic ductal adenocarcinoma or pancreatic cancers. So finally, discuss about how this sialic acid formation occurs within the mammalian cells. In order to for this silations to occur, one of the things that is essential to have is this is the presence of this donor, that is CMP sialic acid, and this is the reactions uh, that is required to form this for that is required for this donor synthesis, that is CMP sialic acid. And after that, this CMP sialic acid can, can donate this sialic acid to any of this monosaccharide with the help of the enzymes, respective enzymes that are mentioned here. That is, in order for this alpha 2,3 silations to be to form at this galactose, it can be formed with the help of this enzyme, that is ST3 gal 1 to 6, uh, and the presence of this donor. Similarly, this alpha 2,6 silations can be formed with the presence of this enzyme ST3 gal 1 2. And similar things applies for this alpha 2 silation and alpha 2,6 silation galnec. So and so this is how the general silation reaction occurs. And the panel B discuss about this different different patient samples that has this pancreatic cancers, to see whether this pancreatic cancers patients has different kind of uh, silation presents or not. And we can see that all of the patients have this donor synthesis presence. And also this alpha 2,3 sialic acids and alpha 2,6 sialic acids are also present in some of the patients. This alpha 2,3 sialic acid and alpha 2,6 sialic acid, and all silations we can see that is present, also present in most of the most of the patients that are affected with this PDAC. And in order to further validate these results from the gene set enrichment analysis, they also did this staining of the uh, pancreatic staining of the patients that are affected with this PDAC with the help of these two lectins, that is MAL2 and SNA. MAL2 can staining of MAL2 will help us to know this alpha 2,3 sialic acid, and staining of this SNA will help us to know alpha 2,6 sialic acid. And we can see that in the compared to the normal pancreas, in the case of the tumors have the more presence of this more staining of this MAL2 and SNA compared to the normal pancreas. So this indicates that tumor cells has more sialic acid presence formations compared to the normal pancreas. And quantification of this is further validate the observations from this staining results, that PDAC has more alpha 2,3 silations and alpha 2,6 silations compared to the normal pancreas. And thereafter thereafter they also validate the silation in the uh, in different pancreatic cell lines, and compare them with the normal pancreas cell lines. In the normal pancreas cell lines, they can find out they can see that only alpha 2,6 silations are present, whereas no sign for this alpha 2,3 silations are present in the normal cell lines. Whereas in the case of primary cell lines and also in the case of commercial cell lines, we can see the presence of both the alpha 2,3 and alpha 2,6 silation acid. And same thing is validated by the flow cytometry, where we can see that alpha 2,3 sialic acids are present in both the pancreatic primary and commercial pancreatic cell lines. So this thing valid these things this the conclusion of these figures come to a point that the pancreatic cancer cells has this alpha 2,3 silations, whereas normal cell lines does not have this alpha 2,3 silations. So the focus of this paper is more on this alpha 2,3 silations than on the alpha 2,6 silations. So in the second case, they decided to check whether these silation data present on the pancreatic cancers are being detected by the Siglec or not, as Siglecs are known to recognize the sialic acids that are present, different sialic acids that are present within the mammalian cells. And based on their experiments, they come to a point that the Siglec 7 and Siglec 9 are known to have shown Siglec 7 and Siglec 9 shown prominent results with the different pancreatic cell lines, as we can see that without neuraminidase treatment the signals are quite high, and after neuraminidase treatment signals are quite low. So these kind of things are observed for the Siglec 7 and Siglec 9, whereas for Siglec 3 and Siglec 10 these things are not been observed. So they from this they made a point that the Siglec 7 and Siglec 9 can recognize the sialic acid that are present on the pancreatic cancer cell lines. So after that, this panel B on panel B is further panel A and B shows the same results, but in the panel but this panel A is shown the results in the case with the help of this neuraminidase treatment and without neuraminidase treatment, whereas panel B shown the results for different cell lines with different ligands in the form of heat map. Thereafter, they decided to check whether this kind of sialic acids are present on which kind of glycans, whether they are N-glycans, O-glycans or glycolipids. So they use different kind of inhibitor to inhibit the glycosylation part. For example, this these enzymes, this kifunicine is used to inhibit N-glycosylation, whereas this benzyl galenic is used to inhibit used to cause this truncated O-glycans, and this PPMP is to inhibit glycolipids. And in this case, when they check this Siglec 7 ligands and Siglec 9 ligands, they can find out that this Siglec 7 ligands are more prominent only in the O-glycans, whereas Siglec 9 ligands are prominent in both N-glycans and O-glycans. And after that, they decided to check which enzymes is responsible for this Siglec 7 and Siglec 9 ligands formation. So they use different kind. They decided to target only in the alpha 2,3 sialic acid formations. That is what they got observations from the panel 1 figure 1. And so they decided to check the enzymes that are involved with alpha 2,3 sialic acid formations. And when they check, they are able to find that ST3 galvans plays an important role in Siglec 7 ligands formation, and ST3 gal4 plays an important role in Siglec 9 ligand formation. And thereafter they validate their results from the already publicly available data of the pancreatic cancer, in which they checked the patients that have both the presence of this ST3 gal1 and ST3 gal4 has the lowest survival rate. So from this they come to they can made a point that the presence of this Siglec 7 and Siglec 9 ligands might lead to tumor proliferation, or prevent the tumors from prevents the tumors from the immune system of the patients. Whenever this ST3 galvan and whenever this Siglec 7 and Siglec 9 ligands are present within the tumors, presents within these PDAC patients and our ligands. So are very whenever the Siglec 7s and Siglec 9 ligands are present within the PDAC patient or in the tumor cells, they are very detrimental to the patients, as can be seen that the survival rate of the patients with this ST3-GAL1 and ST3-GAL4 both upregulated are very low. And then they check whether all the in most of the cells that are found in the pancreas, that which of the cells lines have these receptors, the Siglec 7 and Siglec 9 receptors, and they can see that only the myeloid cells have these Siglec 7 and Siglec 9 receptors. So the further focus is on the myeloid cells. The further focus on the paper is on the myeloid cells, and then they further check whether the Siglec 7 and Siglec 9 signals that they found in the pancreatic cancers are on the myeloid cells or not, by staining the pancreatic by staining the pancreatic cells with Siglec 7 and Siglec 9, and also the markers for this myeloid cells, that is CD14, and they can see that the presence of Siglec 7, Siglec 9 and their markers are almost on the same site. So this further validate the point that myeloid cells are the one that is responsible, that plays a role towards this Siglec 7, that plays a role as a this Siglec 7 ligands or Siglec 9 ligand receptors. So thereafter, once they find out that this the myeloid cells that plays a role in this pancreatic cancer immune evasion, so they decided to characterize these myeloid cells, and when they characterize these myeloid cells, they can find out the different populations of these myeloid cells. That is, some of the these monocytes are differentiated to the macrophages, and some of them are differentiated to the dendritic cells, and some of them remains as a monocyte, and some of them are converted into the proliferating macrophages. And in the case of this normal pancreas, most of these monocytes are present as a tissue resident macrophages, that is TRMAC, that can be seen here. However, in the case of tumors, they are divided into the different, they are differentiated, monocytes are differentiated into the different categories that can be seen here, and also in the case of this map. And in order to the way they characterize these different kind of populations by identifying the markers that are present on this kind of this different kind of different categories of the cells. For example, this monocyte derived macrophages has this kind of markers, that is Marco CD63 and CD68. So they call these populations as the monocyte derived macrophages, and similarly the other populations are also characterized based on the markers that are identified on those populations. And thereafter, what they did is that they again check whether they have this Siglec 7 and Siglec 9 presence or not, and they are able to find both the Siglec 7 and Siglec 9 present in almost all of the different categories of the monocytes that they have identified. And then they check this and they also checked this kind of then they also checked these different categories of the monocyte on the other publicly available data of the pancreatic patients, and they are able to find out that other this kind of categories are also available, are also found in other publicly available data of the pancreatic patients that can be seen here, and that they are characterized in 4 different categories. That is, for example, this monocyte derived macrophage enriched, and this normal pancreatic cells has this uh, tissue resident macrophages, and these monocyte derived dendritic enriched populations are quite low. And thereafter what they further observed is that the percentage of dendritic cells that are present is inversely related to the percentage of monocyte derived macrophages are present. So wherever these dendritic monocyte derived dendritic cells are present in higher amount, the monocyte derived macrophages are present in lower amount. So from these observations, they come to a point that monocyte, there could be a differentiation of monocytes happens either towards uh, this macrophage or towards these dendritic cells. So they use this diffusional map to determine whether this kind of hypothesis is true or not, and they are able to find out that uh, the monocytes are clearly segregating into the two different populations, that is monocyte derived macrophages and monocyte derived dendritic cells. So and uh, thereafter, they also decided to check what is the impact that has been observed on the survival probability of this different kind of populations. That is monocyte derived macrophages and monocyte derived dendritic cells, and they are able to see that the patients that have high ratio of these monocyte derived dendritic cells to the monocyte derived macrophages has higher survival probability compared to the case where the monocyte derived macrophages is high compared to the monocyte derived dendritic cells. So from this they made a point that monocyte derived macrophages plays a role towards this tumor proliferation or tumor helps the tumor cells from helps the tumor cells in immune evasion, whereas these monocyte derived dendritic cells have this as some kind of anti-tumor properties. And thereafter from the previous figures, they are able to make a point that monocyte derived macrophages this has the properties that supports that helps the tumor cells to grow, whereas monocyte derived dendritic cells has the properties to suppress, has the anti-tumor properties, and these things are been further validated with this statistics data, in which case they find out that the percentage of cancerous cells that are present in the case of this monocyte derived dendritic cells are very low compared to the case this where this monocyte derived macrophages where percentage of cancerous cells are very high. Similarly, the percentage of normal duct cells are very are normal duct cells are low in the case of this monocyte derived macrophages compared to the case of monocyte derived dendritic cells. So from this results and from the results of the from the analysis of the previous figures, they made a point that it could be the tumor cells that might helps in differentiating these monocytes into the macrophages, so that these macrophages can help prevent the tumor cells. So considering this hypothesis, now they did some algorithm to identify uh, they did some in silico analysis to identify which are the genes that are responsible for the differentiating of the monocytes to those macrophages, and from their in silico analysis they are able to find out that this CSF1 gene could be the is the highly responsible is could be the gene that is highly responsible for differentiating monocytes to these macrophages. And in order to validate this in silico analysis, they did this co-culture experiment of the different the different pancreatic cell lines with these monocytes, and they are able to they use these different kind of pancreatic cell lines with the monocytes, and they are able to cluster the cells into different populations, that is tumor associated macrophages and CD163 low monocytes. So in this analysis, they considered this tumor-associated macrophages that has the CD163 markers, and the cells that does not have these CD163 markers are considered as this myeloid population, this CD163 low population. And similarly, they also characterize these cells with this mannose receptor, high mannose receptors and low mannose receptors. And the further analysis they did of of the results from this co-culture assay is about this: what are the percentage of CD 163 cells that are present uh, in this population. And they have find out that in the case of this PL 45, the percentage of CD163 cells are found are very low. So this could indicate that in the PL45 cell lines, formation of this macrophages is quite high, as the CD163 is the marker for tumor associated macrophages. So in all of the cell lines, the TAM formations are occurred, whereas in the case of PL45, the macrophage formations is occurs. So they further validated these results using some this dimensional reduction analysis, and they can see that the monocyte is differentiated into this TAM, and also this and also into this CD163 low, or these macrophages. And some of the monocytes are also differentiated towards these dendritic cells. So these things, uh, and this is the analysis in terms of the CD163. So this CD160 high CD163 is the marker for this tumor associated macrophages, whereas low CD163 is the marker for low CD163 is considered as the macro is considered as this monocyte derived macrophages. And and uh, uh, after this co-culture assay, they decided to again do the similar kind of experiment by in vitro differentiation through different through other techniques, such as this transfer techniques, or this by inhibiting this CSF1R receptors. And in this case, they can find out that in the case of this transfer in vitro differentiation, with the help of this transfer uh, techniques, they can see in the case of this BXP3 and other cell lines, they can see almost comparable formation of the CD163 cell lines, whereas when this CSF1 receptors is inhibited, the formation of this CD163 cell lines is reduced. However, it is not completely reduced. This indicates that it is not only the CSF1 that plays a role towards this TAM formation, whereas there are other factors that could also helps in the formation of these tumor associated macrophages. So so far they made a point that it's the tumor associated macrophages or monocyte derived macrophages that helps the tumor cells in immune evasion. That helps the tumor cells in immune evasion, and also this tumor cells helps this monocytes to differentiate into this monocyte derived macrophages. And this CSF1 gene also plays a role towards this differentiation. Thereafter, they decided to check whether it's the sialic acid that helps in this differentiation, or it's something else. It's So they decided to do this experiment. They decided to check whether the sialic acid contributes to the differentiation of the tumors to monocyte derived macrophages by interacting with the Siglec 7 and Siglec 9 or not. So in order to check this assay, they first check the supernatant of these pancreatic cell lines, whether they have any Siglec 7 or Siglec 9 ligands or not, and they can see that these supernatants of these pancreatic cell lines has the Siglec 7 and Siglec 9 ligand. And also the presence of Siglec 7 and Siglec 9 ligands is also validated using this flow cytometer reading. Thereafter, they check the Siglec 7s. They check the interaction of the Siglec 7 and Siglec 9 by introducing the uh, by blocking the Siglec 7 and Siglec 9 receptors. So, and they can see the formation of the CD percentage of CD 163 plus has been reduced whenever they introduce Siglec 7 and Siglec 9 inhibitor. And this could indicate that Siglec 7 and Siglec 9 are the one that plays a crucial role towards this macrophages differentiation. And they validate the same thing using this by knocking out this donor, by knocking out this CMP sialic acid donor, by knocking out this CMAS enzyme that helps in the formation of the CMP sialic acid donor. And this is how they validated their knockout, where the knockout has no sialic acid, whereas the mock has the sialic acid presence. And when they check when they check for this CD163 and CD206 markers, they can see that some formation of this CD206 and CD163 uh, they can see that some reduction in this CD206 and IL-10, whereas the CD163 formations is not reduced. So from this they come to a point that this this sialic acid along with Siglec 7 and Siglec 9 is essential for this tumor associated macrophages differentiation, as it will not only helps in the tumor associated macrophages differentiation, but this immune suppressor indicators such as CD206 and IL-10 will also be produced when will also be produced in these tumor associated macrophages whenever this sialic acid and this Siglec 7 and Siglec 9 are all of them are present. Only this Siglec 7 and Siglec 9 presence will not help in the formation of tumor associated macrophages that can have these immune suppressive properties. Thereafter, they decided to check. The whether it's the sialic acid that helps in the differentiation of these monocytes, or there are some other factors. So they use these sialic acid dendrimers. They co-culture these sialic acid dendrimers with this monocyte for 4 days and then check the monocytes for the different markers, that is CD163, CD206 and PDL1, and they do it in two different conditions. First one is this MCS CSF, where there is MCSF presence and there is MCSF is not present, and they can see that when this MCSF presents, the formation of this presence of these markers are increased compared to the case with MCSF is not present. So this substantiates the point that sialic acids are the one that helps in this monocyte differentiation. And after that they also check the other markers, that is checkpoint inhibitors such as this interleukin 10 and interleukin 6, and they can see that whenever this MCSF are present, in the presence of sialic acid, the formation of this IL-10 and IL-6 are increased compared to the case where this MCSF is not present and sialic acid interleukin 10 formation is low, whereas there is not much effect on this interleukin 6. So this interleukin 10 formation is affected by this MCSF presence. And also they check whether this kind of monocytes are been detected, are been recognized by this Siglec 7 and Siglec 9, and they can see that the Siglec 9 can recognize this kind of monocyte derived macrophages compared to the case of this Siglec 7. And thereafter they decided to check the tyrosine phosphorylation of this kind of macrophages, tyrosine phosphorylation of this monocyte derived macrophages using Siglec 7 and Siglec 9, and they can see that when it's compared to the case of Siglec 7, in the case of Siglec 9 there is an increase in the signal. So they can come to a point that it's the Siglec 9 that has caused this helps in the differentiation of this monocytes to macrophages than this Siglec 7. And finally it is decided to check whether this sialic acid diatomers can further differentiate these monocyte derived macrophages towards this TAM, that is tumor associated macrophages, or not. And in this case they use these monocyte derived macrophages and co-culture them with pro-inflammatory signals, such pro-inflammatory agents such as LPS, with the help along with this sialic acid dendrimers, and then analyze using FACS and ELISA, and they can see that there is a upregulation of the indicators such as this PDL1, and also this interleukin 10. However, there are some indicators that are reduced, such as this some of the immune suppressors are reduced, such as this HLA-DR and this HLA-DR and this LTA4, and also this TNF alpha, which is one of the immune suppressors, are also reduced in the case of this pro-inflammatory agent, that is LPS. So this this further made a point that this sialic acid can can cause the immune suppression also in the case of this pro-inflammatory conditions, such as in this case in the presence of this pro-inflammatory agent, that is LPS. So this further made a point that this sialic acid could differentiate this monocyte derived macrophages in a way that can help the tumor cells in immune evasion. So what this finally summarize, what's the final summary of this paper is that these monocytes have these two kind of receptors, that is Siglec 7 receptors and Siglec 9 receptors, and when they infiltrate at the site of these tumor cells, that sialic acid that are present on the tumor cells can differentiate those monocytes into the macrophages, and these macrophages then can help the tumor cells in immune evasion and promote this growth of the tumor cells through by upregulating and downregulating these factors that are mentioned here. So that's all for this paper. If you have any questions, feel free to comment. I will be happy to answer your questions. If you need to understand more about this paper, you can go to reference that has been provided in the description. That's all for this video. Thank you for your time.